So I find great pleasure in welcoming you to the 8th Abdus Salam Memorial Lecture Series organized by the Syed Babar School of Science and Engineering. This lecture is Pakistan's only Nobel laureate in the sciences. The speaker for last year was, in fact, another Nobel laureate, Professor Donna Strick. Undoubtedly, we have an equally distinguished speaker today. I find great pleasure in introducing to you Dr. Shahid Khan, a biophysicist, a neuroscientist, and he's made lasting contributions to the Pakistani academia and biology, which is an esteemed, distinguished research center and teaching and learning center in the Punjab University. And he helped set up this institution. We are sitting or standing today. He was instrumental in the design of the Syed Babar School of Science and Engineering, its curriculum, some of them have become tenured faculty members. He's mentored numerous students. Some of these students have now found place in the best institutions all around the world, and later he completed his studies at the King's College, Yale, and Caltech, where he was a postdoctoral researcher. Later on, currently he's a senior scientist at the Lawrence Berkeley Lab, and his work spans over decades. I think that his own career has progressed proteins, and proteins not just as static structural edifices, molecules, but proteins that move, proteins that are dynamic. Welcome, Dr. Shahid Khan.
To be here, this is almost my second home, certainly in terms of, my scientific family. In fact, for my scientific family, I will sketch my own life and how I developed, and I learned a few things for which I'm grateful that have made me appreciate how this world is made. Than me, some of their insights and absorb them, and then lastly, I've been most fortunate to have good students. So I will tell all of these things. So Mahajir parents, and actually double Mahajir parents, because my father was originally from Dera Ismail Khan and my mother's family is originally from Mosul in Iraq. But then they migrated to Hyderabad. The Muslims had become very bad during partition, and they moved to Pakistan in 1961. So this is me at around, I guess, 7 years old. In 1961, that's when I enrolled. It didn't go too well. The headmaster told my father, he seems a little bit slow to comprehend things, but we'll put him in the B stream and see how he does. So not exactly a good. I want to say, it turns out in this time, while I was a kid growing up, some of the seminal work for molecular biology was done. So the first thing was could be deconvolved with physical methods to build a model of DNA using the chemistry known at that time, but equal ratios so the chemistry supplied the base pairing aspect, and the diffraction pattern supplied the structure, the hereditary material. Now, so of course, DNA is very important, but what really makes the cell are molecules called proteins, or proteins where these are a distinct substance. But beyond that, there were various speculations. Well, could this be some kind of an amorphous material? Did it have any structure? Actually, in 1958, Sanger sequenced insulin and showed that protein was a linear combination of distinct amino acids in a protein, and they're arrayed as a 1D string where each building block is connected to another building block by a bond. Interesting and complex is that these building blocks, the amino acids, have different side chains. So some of those are oily, some of those are result, as we shall see in this lecture, in a lot of different properties. So the first thing is, all right, you have a linear string, but then various hydrodynamic measurements and so on were globular. So how do you get a spherical particle from a linear string? Well, that was resolved by diffraction studies, where actually you went indexing and phasing the pattern, you know, I can if people have questions, I can answer those, but I don't want to get into the details. String folded into very distinct and beautiful architectures that were alpha helices, rod-like things, or beta sheets that folded in globular proteins. We're talking of protein in solvent, which is water. So the organizing principle for the protein is that it's an oil drop and it's covered by a polar shell. And this was one of the first with Max Perutz, who got the Nobel Prize for hemoglobin, which people may or may not know this, took him 27 years to solve. So it's important to know we have proteins, and we know that they are a globular thing. They have a particular architecture. Now I am going to touch on how does it do work, and proteins do all kinds of work, but I have tried to understand proteins doing mechanical work, and the reason for that is that mechanical work, you have a good measurement of the work done, and then you have to understand the energy input and the energy currency, which I will talk about. So that was the early phase of my career, and then I got interested with proteins that switch and are responsible that work as motors. There are proteins that work as switches, and then these motors and switches form computational circuits. And this has been the last period of my work. So and again, I will not talk about any of the technical details, but what I will do is in this talk, there will be, I have referred to my publication, so people who want to go and really read about this publication. So my first contribution had, and that was made when I was an undergraduate at was solved, and again this is kind of interesting because I was talking to some students who were saying, well, sir, we don't really know what we are going to do. When you get the right signal. So in my case, I was actually so first of all, I survived Karachi Grammar School. That was my first. So I had gotten a scholarship as a physical chemist to study physical chemistry at Imperial College. And I said, "Theek hai, I will go there," but I also applied to this funny program, new program called Molecular Biology. I had no idea what molecular biology was, but I said, "Theek hai, I mean, you know, I'm going to do chemistry, but let me go and give an interview to the Huxley, who at that time had done some microscopy to understand how muscles work, and Hansen and Huxley have a famous model future not only of biology and of chemistry. But I was still nervous. I said, yeah, but I don't know anything about it. So, but she said, don't worry, I mean, you'll find out. I mean, you know, if you think it's student, don't just box yourself into this or that. I mean, you know, you'll find out. Just be passionate about what you're trying to do. So let's get back to the science. So we know change shape, and how proteins change shape is a very important issue that has been worked on for the past half century, and we are learning more and more about it. the and Shanghai, where they basically they by various elegant studies found that you had proteins that could bind and to expresses a protein. It also does other things, but for our purposes, gene bit of DNA expresses a protein, but it's controlled by pieces of DNA in front of it that determines whether by another substance, in the case of the lactopteron, by lactose, and a compound that mimic lactose, it comes off an expression different place on the protein than the DNA. So there must be some kind of change and signaling within the protein molecule that's happening, and how is that, which is an enzyme basically which puts a phosphate on glucose. So this protein, the way it puts the phosphate on is that it changes shape and hugs it, so that the phosphate, the glucose is buried and is in contact with ATP, which is the substance that says you have hexokinase. And then this was for a globular protein that's in water. So now the next thing was, one big idea is that all biological material is made up of cells, fundamental units that are there in bacteria to humans and make up, but the cytoplasm, and they have membranes. And these membranes are essentially double layers of lipid. They are like insulators or capacitors. But one of the important functions they serve is to transport ions and work as pumps. So this is a very simple pump called bacteriorhodopsin, and this was showed. So they couldn't solve the whole structure at that time. They found that but these membranes, the protein is in membranes. I do a projection that's what it looks at projection. And actually, it took this group 15 years to get the final 3D with other with a team in my school. First at King's College and then moving to Yale. These are these alpha helices that I talked about. How do we fit the amino acid sequence, the chemistry of this protein, the one-dimensional string, into the three-dimensional way that we could deuterate specifically a particular amino acid and then we could essentially by doing diff protein and then do it again for another deuterated amino acid and so on, using neutron diffraction. So the neutron diffraction is because X-ray scattering factors have to do with interacting with electron clouds, whereas neutrons interact with nuclei. And of course, if you deuterate, you're going difference in the scattering. And so we use that to assign now the chemistry. So we had a chemical model now of the structure, which was subsequently proved correct. It would be to ask the question, okay, so this is proteins have this property and we can energy input. So I had talked about this substance called ATP. So ATP is your energy currency in the cell. It's like the petrol that you use to cells. But then the question is, how is ATP made? So the initial source of energy comes from the sun. You have light, and that uses the energy from light to make compounds ultimately. Or if you're an animal and you cannot use light like we are, then you have to take in food. And then the ultimately of taking in food is that the food is broken down, and then you there are proteins within by a series of reactions separate charge and maintain a potential across membranes. So essentially, and the whole process of charge set I will not talk about, but what I will talk about is, and it was known by the time I got interested in working on this, people could insert electrodes and actually measure potentials. But ATP is made in small organelles called mitochondria or chloroplasts in plants, more complex cells. And these bacteria also have, the logical deduction should also have potentials across them. And that was a conductance and so on of various ions, and that the relevant potential to make ATP is the proton potential, both the electrical problem. How does one measure this? So the problem is that if you have something like a mitochondrium or a chloroplast or a bacterium, you can't really. That was to use radiolabeled cations that would partition across the membrane, or radio-labeled weak acid that would part. Biochemist working at the Roche Institute, their R&D department, we got those from him, and we, by measuring the radio-labeled distribution, we of accuracy. And then I chose to actually relate it to work, as I said, by choosing a system where one can measure the mechanic is a rotary motor that actually works by rotating. It can rotate as fast as 300 hertz and even faster membrane. Now, and we then did some studies which I will not go into, but basically our measurements provided evidence of a Brownian random walk, if you like, of particles in the membrane proteins that had a certain elastic compliance, and that would then Wolfgang Junger, another a colleague in Germany, showed that that mechanism actually applies also to how ATP is made. And protons flow through the membrane part of the ATP, rotate this thing, and that then forces, in this case, so that's the mechanism whereby this energy currency ATP is made. And so that was the measurements of rotation and the structure of ATP, like Wolfgang and so on. So that was one issue that was important. Then the other issue was, all right, so we can measure work a lot, but there was a problem, and the problem was that the bacterium, to study it, we were studying, so it was difficult at that time, so the cell rotated instead. But now we are talking under a high load condition, so everything was twisted, and so that twisted thing acted various reasons. Then we cannot measure the actual mechanics with the precision we wanted to. So a big thing in the mid-80s now, so this is incidentally the lab where the first action potentials were recorded in squid giant axons. And I went there because for a project that didn't work at all. I had potentials across electrically across bacteria. Now, and that was inspired by the fact that for sodium channels. But this did require, so the one thing to note again here is so the fastest channels like sodium channels and so on would pass about 10 to the 9 ions per second, and proton channels are even slower. They're about 10 to resistance. You're not going to measure a single channel conductance. So my project well, it failed because of that, and also because I hadn't realized, I did not fully learnt or appreciated to really take off the cell wall, because bacteria have an outer cell wall, and so if you do not take that out, so it was a bad idea, had really no chance of working, but I learned a lot. That was the first thing. So in that sense, no idea is really a bad idea if you learn from it. And then the second thing is sheets, and Mike Sheets had shown, working with Spudik at Stanford again around 1984, that if you essentially you can attach a bead and monitor the rotation of the bead on them, and you can do that if you use a CC properties of the camera. So firstly, it's a digital signal. The signal goes on, is recorded on pixels, so you can analyze. Contrast transfer function, so a camera can actually see things that the naked eye cannot. It has, you know, and again we can talk about that, but by that tubule motors, how do they move? And now, of course, everything at the nanoscale is dominated by Brownian motion, but spread it out would be moving this way and that. You know, it would basically walk like a drunkard. But there's another motor, kinesin, which basically would walk along a protofilament. Kinesin is a motor that is used in axons to transport axonal vesicles, you know, long distances, as much as a meter in the just to quickly summarize this section, what I hope I have given in this first section is to give some, you create a proton potential that then makes ATP. Proteins are oil drop, everything is dominated by Brownian drift. These motors, by the way, so we then man-made machines, and so really understanding how they work could help us make better machines. I think they're effective. And but you in the case of the proton transport channel, the protein basically couples the proton flux very tightly because of its ability to change. So it's a very high coupling. You know, there's no waste. There's no sort of generation of heat as such. So, and then the other thing just to note is that motions of molecules, as we did and as many others have done, is because inertia is negligible. So people who know fluid mechanics know of a number called the molecule. That ratio, the value of that ratio is as if you were swimming in molasses. So there's no coasting. The moment you stop exerting force, you stop. And so that's again, so optical microscopes measure motions, so they can't resolve because in the optics, the resolution is limited by the optics, by something called the point spread function, which again no limits to the precision by which you can measure position, and you can actually measure that with nanometer precision and millisecond resolution. Again, much better than we humans can do, more than a millimeter. So the thing is that this is a studying molecules, you are in a different world. You are in. Just to go very briefly now is to talk about how do you build a switch. So this is work that I did. So this is me as a associate, well, Illinois, and that had to do with measuring how bacteria sense food. So that a bacterium would swim and come together to form a bundle that pushes it, but then it changes its rotation sense, so the bundle flies apart, and that then tumble behavior is important. Why is it important? Because the bacterium is very small, which means it's being bombarded and it's losing course. So it cannot keep straight normal motions around it. So this is actually turns out to be a very interesting strategy and a very efficient strategy to migrate towards the source of food. And that happens, and one can show that by suddenly changing the environment. In this case, we did it by photolyzing a amino acid, a food for the bacterium that it likes, in a straight line after the stimulus, which is, so we coupled it to with a protecting group, so when you shine in near again it would. And the important thing to note, well, two things, is that this is mediated by, well, so the food has to be sensed, so there's a receptor, and you have the one protein that changes shape when it binds a phosphate, is modified by a single phosphate group. So in this case, so phosphor, two Nobel Prizes that have been awarded for this discovery. First, that protein phosphorylations are important, and then receptor responses in the cell. So using these methods, we showed that chemotaxis is very sensitive. Still, think about even now. And then the other thing which has to do, I hope this is in neurons. So this is a neuron. These are a neuron from the hippocampus, which is an area of the brain that senses memory, and or basically is a firstly communication between neurons. So if you have a particular neuron that has little spots in it called dendritic spines, which communicate across a gap called a synapse, where the so you have an electrical signal, then you have a chemical communication of memory. The important thing was an important idea is something called the Hebs postulate. And what is Hebs postulate? Hebs postulate stores memory. The other spines here, other structures that are not in contact are not affected. So how is memory stored? So, well, when you have stimulation here, you have a neurotransmitter across the synapse that then releases, results in a pulse of calcium that then results in increase of the stimulated spine that grows in size. So actually some measurements to confirm some of the fundamental tenets of it, and the basic take-home message is that the calcium is binding, then comes off the actin cytoskeleton. These are stringy things that are, by its name, are the cytoskeleton inside the cells. Actin essentially measure all of that using optical techniques. So oops, I don't need to do this, I just need to go on. Methods where using again a branch of microscopy called total internal reflection microscopy actually measure the association of single molecules of this thing. Go into the details of it just to give you an idea of what can be done. So this is now actually meeting the middle phase of my career. So again, just to summarize that briefly, what that middle phase, these are the sort of key as sensory cells, but they are sensory cells. They can actually, they need to store some information and remember it in order to chemotax. Neurons, of course, have long-term memory, but the initial mechanisms of memory storage all happen within the first minute or two. They really do involve molecular events that give specificity to the compartment protein assemblies, So in this case, you have the calcium calmodulin kinase, which is like a pinwheel. So it is holding many actin filaments together. It comes out, and the rest is sort of self-explanatory. So I think I will just finish latest work, which has to do with since I moved back from LUMS, and this is my work is because again of a radical change in the scientific environment that has to do with the fact that bioinformatics is we talked about the early protein structures, and if you track structures by year, you see that the growth, there were only 13. This is, in 2020. So now we have a large database, and we can now use that database to learn and predict protein structures. Use that test various algorithms that can even predict proteins, and so in 2020 there was DeepMinds and some of this, and indeed my work that Sabi mentioned in Punjab was to set up the bioinformatics unit in Punjab, because and I'll in the acknowledgements I'll say how I collecting sequences, but then how to analyze them and to make sense of them was something for which you needed bioinformatics. And so that's one thing. Only conserved residues that might be important for function, but also co-varying residues, and by that you could actually also map it onto dynamics. And it turns out, again, I will not go into any detail, but these are the papers, that a trace of what functional protein motions are important and retains the context for those functions. So that's one kind of thing that I am now pretty excited. Whole expansion response with accuracy as basically as a electrical engineering circuit. I mean, people who are electrical engineers would appreciate this, and ODE analysis, things that engineers can do to fit the data quite well. And I will not go into this. And the final thing, structure of CAMKey2 that I have been working on with some colleagues, Professor Vachu at Stanford, which is allowing us to not only see structures and so on that could give information on variable subunit stoichiometries and how they might relate to function. And this is so small. In fact, आप तसव्वुर भी नहीं कर सकते how small, 30 nanometer. But to actually measure and define this molecule, this is what you need. You need the seminal work in electron optics by Ruska in the 1940s and 50s, then with techniques to enable vitrification, freezing, direct electron detectors, as opposed to CMOS cameras and so on, which would essentially measure photons via the photoelectric effect. You can now have direct electron. This machine that allows us to do this. This is some of the things here. One of the important things that I will well, it's not really noise. I would basically, noise is when you don't understand something, we call it noise. But actually, it's variability, which in itself gives us information. So let me finish my personal journey, and I want to thank. So first of all, I thank the Almighty, because I've been blessed in learning some features of how this is Salam Saab. So when I was a postdoc at Caltech, that's when I met Salam Saab, and I told him, look, Salam Saab, what can I do? I mean, he said, well, don't worry about that. I think, you know, there were, you have to get people interested. and he said, okay, look, what you should do is there's somebody to gain the support, work in the Tata Institute of Fundamental Research, and was successful in attracting some really good Indian scientists back into India. So talk to him. And so, you know, I would certainly acknowledge these had a big effect in shaping my own career. And these are some of the other people. Peter Mitchell of the chemiosmotic hypothesis, Tom Stites, who of the hexokinase thing. So he's a Nobel laureate who got his DNA into RNA, the first step in making proteins. I haven't talked about this. John Walker, the person, the Nobel laureate who solved the Wolfgang Junge, who established the rotation, proton-driven rotation, basically the model that Howard and I had proposed for the bacterial motor. A chemist who again has you know, helped me synthesize and worked with me to synthesize all wonderful organic compounds that also helped us in particular in LUMS, because Mike Sheets then was set up at the Mechanobiology Institute in Singapore with the support of the Sheets when we were setting up our own school here. And all of those students now are in the US, but they are they benefited enormously from the mentorship. I acknowledge my students. So a number of them are professors. I mean, we have Professor Iram Liaquat at Government College here, who was with me here at LUMS that I had here. I mean, I had outstanding students in the USA, but I'm particularly proud of the students that I had here. This is Shafqat Rasool, a student. He is at 2013 batch LUMS biology, and he will be giving his thesis defense at McGill University this month. And Shafqat will also be to be held in March. And he's played a very active role. So he, together with a lot of my students, have formed an association to help people. So we are looking forward to you visiting us, and they would love to be involved again in LUMS. So you know, that's a special shout out that I want to give to Sabi, and thank you so much for listening.
The floor is open for questions.
May I?
Ji.
You know, you're talking about protein. Where does protein play?
आपके जो stomach में digestion है, that's all proteins. And it's the structural compound, you know. It's in every made up of proteins. Proteins are the workhorses of the living organism. और the work on motion, इसका immediate application जो मेरा काम है, has to do with using ATP that I talked about, energy currency है ना. But if you want to change its ability to bind ATP, you don't design an inhibitor that competes with ATP. So, मगर if you know कि you can bind, okay, this part of the protein is communicating, its motions are communicating with how this active site is against that protein, it's only opposed to your whole other proteins. So specificity drug design में बहुत important है, vaccines develop. फिलहाल इस वक्त I'm in the division of material science in Lawrence, Berkeley. I mean, I'm not in the biology department division. In fact, they don't really have, you know, but Lawrence, as Sabi knows, Lawrence Berkeley make better materials. So yeah, so these are all the things. Ji.
First of all, thank you very much for a wonderful. My question is there any difference between the dynamics of enzymatic or functional proteins and the structural proteins? And if they are different, then what?
We need to answer that with a broad brush. but essentially जो आप तो एक extreme मिसाल देंगे, the proteins in your hair cells, so the cell का structure for years, agar if they don't pair up, you'll become deaf, but you don't become deaf for a long time. So they are basically locked in a crystalline structure and their motions are frozen. That have to do with you could have either very subtle, so hexokine is an enzyme that has large motions, magar reaction, you are not going to change the free energy of the reaction, jaise aapko pata hai, you need to change the activation energy. Or वो activation energy change करने के लिए you basically by either जैसे hexokine is folding over and hiding the the compound that you want to catalyze from solvent water, of electrostatic side chains, polar side chains that just form a very precise electrostatic change. So in both cases protein motions are involved, but they are components that capture collective motion, the high eigenvalue eigenvectors or principal components. In the other case there would be the smaller principal components, because but that is why it's very important to understand motions. So hum log, one of the things that again I did not, well, there's a lot of things and a lot of wonderful colleagues that I have not mentioned. One wonderful colleague was Safi, we've realized कि वो ये protein में जो pockets होते हैं, they just bind water, because protein by and large bridges and so on, that again can affect catalysis or other functions. Ji, Shahid.
Yeah, thank you very much, Dr. Shahid, for overview of your career and this
So I actually realized that when I was preparing for this talk.
So with this development of technology in the proteins, when we talk about functions, molecular recognition, which dictates certain functions, so proteins are important, but lichens,
right.
are much more flexible and much more diverse in terms of the structure and dynamic properties, play more important role.
Yeah, there are a lot of others. So ye bilkul sahi hai. And then as the biologists know, RNA is now are an important, RNA things. So there are now RNA machines and so on. So we are at London, and he is a theoretician. Or usne so we say, we've got these hundreds and thousands of structures, but when you reduce them to common folds, they're not so many. Conformational spaces of protein folding, how many different folds can you get, and what fraction can you estimate we have discovered by now? And the answer is about 10 to be discovered.
Actually, dynamics is really important. I generally give example to students in my class that when we look at structure is important, but when we look at antibody, a simple scaffold, all those non-structural flexible parts.
No. I mean, that's why I spent most of my life studying it. Bilkul. Can give is and this actually is that you must dare to dream, or in fact why this is important. Institute for Theoretical Physics in Trieste, the reason was that he said it's not just only the all levels, bilkul ala science, the science belongs to everybody, and it is the right of everybody, regardless of whether they are in a developing country or not, to do science. Or ye mujhe was shaping up as a kid. I had long, an angry argument with my, Walid marhum, and I used to, become an engineer or a doctor and provide for your family, ये क्या कर रहे हैं, ये तो Germans or Americans के लिए है. And I think that this is something that is probably just follow what excites you, because that is the way you will be successful. Doesn't matter what it is, but if you follow what excites you, you will contribute to और आपकी शादी भी हो जाएगी and all of this. But this is, do what really excites you, because the greatest wealth that we can, then knowledge. So follow what you feel excites you in gaining more knowledge. But जैसे मेरा example मैंने आपको बताया, वो resolve होगी, but other areas turn up, right? So now computational biology has become an important avenue because it can teach us. So basically, those are the two important things. Ji.
Thank you very much for a very nice talk. So when you were mentioning about ATP being the energy currency, I've always wondered why evolution.
So ये अच्छा question है. I don't know the answer to that. People are trying to there are people, these are experiments where you simulate what you believe was the primordial environment बिल्कुल शुरू में, and what kind of compounds you would form, and ATP भी होती हैं. And remember, for making DNA, replicating DNA, you need the triphosphate things of all, right? So, but people is something that we could, because I would not, you want to, I think it's an important thing to think about. I would not have the answer to that, but जी, हाँ.
Any questions from the online forum?
हाँ, अभी नहीं, all right, okay.
So, adding to what Abdullah said, we reside in the physical world, right? So, when in a certain environment, a certain molecule is introduced, it conforms to the surroundings, right? It moves that, or charge densities on the surface. So, I think maybe ATP was more feasibly able to transform into its other form, random gas. Maybe that was more feasible physically for it to go to that form, and we can maybe use stochastic self-assembly or learning algorithm using neural networks or stuff like that.
इसका ये है कि तो science के one is to there are two things that kind of help me when so one is to have ideas. So just you've had thing that we could do to sort of rule out other ideas. So and so both are important. और ये learning का so I would just caution in deep learning and things because of, but the learning these learning algorithms are as good as your training set, right? तो अगर आपके पास अच्छा आपके साथ बात भी हो रही थी in predicting single domain proteins ये वो, मगर ये multi cellular assemblies जैसे मैंने बताया in the last slide of this ring structure and how और सफ़ी जी.
has been able to hire so many fabulous faculty, and the culture that was set up by Shahid Khan, we still feel it, although he's not here in Pakistan with us, not be having vocabulary, we thank you enough for that. So God bless you for that, and this is also for the students, because now we are actively collaborating on you, especially the protein structures and how X-ray mass spectrometry works for us for structure determination. So I'd like to thank you on every year and learn more from you. I had a question, but I wouldn't ask it in conversation.
ठीक है, नहीं वो चलें, but I mean I say well thank you, and I certainly do intend to straight always. It's a two-way street.
So
So, by the way, the question that you asked has also, in fact, engaged Abdus Salam as well. He has an obscure paper in an obscure journal, particles and
Chirality, yeah, right.
antiparticles,
Right.
matter versus antimatter. Why do we prefer a particular chirality? So Abdus Salam called this symmetry breaking, on symmetry breaking. But these are open questions. So without further ado, Safi has given a great epilogue and a great vote of thanks on behalf of all of us. It's hard to again in
हाँ बिल्कुल बिल्कुल